All right, welcome to our notes on the LacOperon. Before I jump into the notes, I have to explain a few things and then we'll get going on it. First of all, the concept that we're trying to get is something called regulating gene expression. What that means is, and I'll read the note that says here, the cell doesn't want to waste energy building proteins it doesn't need, so it has mechanisms to turn genes on and off. The example here is called the LacOperon. This is a very simple gene found in a bacteria called E. coli. The human mechanisms for turning genes on and off are actually somewhat more complicated, so this just gives us an introduction. Despite me saying that it's simple, you're going to find it's fairly complicated. First of all, the LacOperon is a series of genes whose job it is to digest the sugar found in dairy products, lactose. That's where the word lac comes from. And operon is a whole bunch of genes that work together. Now, if the E. coli, if this bacteria is floating in a dish of milk, it wants to digest all the lactose that it can. However, if there's no dairy product available, if there's no lactose in the environment, it would be wasteful for the bacteria to be producing the enzymes that digest lactose. So it has a way of telling if lactose is present in the environment, and then if it is, it turns the genes on that allow it to digest lactose. And as soon as lactose is no longer in the environment, then it can turn those genes back off again. Okay, to understand how the LacOperon works, there's a whole bunch of vocabulary words, about 6 or more, that we need to go over. But as we go over the vocabulary words, you'll start to understand how exactly it is that this operon works. So the first word we need to know is the word operon. Operon simply means several genes that are transcribed together. So in the diagram, starting where it says DNA, and the series of boxes moving across to the right, those are the genes that are transcribed together. If you notice, there's a circle up above, that's RNA polymerase that wants to read the gene. Now, the structural genes are genes that specifically code for a protein, but some of these genes, like the one that says PLAC, and the red one, that one was orange, the red one that says O, they don't actually code for a protein, so we don't call them structural genes, but they are important, and you'll get that in just a second. There's a repressor. A repressor is a protein, and that it looks in the diagram, it looks like a blue Lego piece. The repressor is a protein that attaches to the DNA and stops the RNA polymerase from reaching the structural genes. Now let me make that make sense. The repressor I sometimes refer to, because it starts with an R, the repressor is a roadblock, and it prevents RNA polymerase from moving ahead. So it snaps onto that red O, and then RNA polymerase can't unwind, unzip, and read one side of the DNA. It can't get to those 3 green genes where it says LAC-Z, LAC-Y, and LAC-A. It can't get to those because the roadblock is in the way. That means, in essence, that there's no lactose present, and so we don't need to get to those 3 genes. Now, the operator is that red switch where it's called the on-off switch, where the repressor sits. So the blue Lego piece, the roadblock, snaps on at the operator and it prevents RNA polymerase from moving ahead. The promoter site is the orange area right before the operator. It's a gene that does not code for any proteins, but it's where the RNA polymerase sits when it's getting ready to transcribe the gene, when it's getting ready to read across the O, the LAC-Z, the LAC-Y, and the LAC-A. The regulator gene comes before it somewhere else on the DNA, and that's the gene that actually codes for the repressor, the roadblock. The roadblock has to come from somewhere, and so where does it come from? Well, there's a gene somewhere else in the DNA that produces this protein that then snaps onto the operator and stops RNA polymerase from moving ahead. So now, how is it that RNA polymerase ever gets to move ahead? Well, you have to remove the roadblock. So you remove the roadblock with this purple or pink circle on the diagram. That's called an inducer molecule. In this example, the inducer molecule is called allolactose or lactose. You could call it either one. The inducer is a molecule that causes the repressor, the roadblock, to change shape. And when it changes shape, it no longer fits like a Lego piece and it falls off the DNA molecule. Once that roadblock is gone, RNA polymerase now can move across. So the idea with the LacOperon is that the roadblock is present until lactose shows up. Once lactose clicks onto the repressor, then RNA polymerase can read across and it creates the mRNA and eventually the proteins for digesting lactose. All right, now that we've gone through the vocabulary, we've pretty much explained how the LacOperon works. So imagine a bacteria that's floating in a dish of water. So lactose is not present. There's no lactose sugar in the water. So we don't want to waste energy building the proteins that help the cell to digest lactose if there's none present. So the operon is turned off. That means that the repressor protein, the roadblock, is sitting on the operator gene and it is blocking RNA polymerase. So RNA polymerase can't go across and read what here is called gene 1, gene 2, and gene 3. Okay, so now if we picked up that E. coli out of the water dish and placed it instead in a dish of milk, lactose would be present. So the first thing that's going to happen is that lactose or allolactose is going to click onto the repressor, and that causes the repressor to change shape and to fall off the operon, or I'm sorry, off the operator. That frees up RNA polymerase. There's no more roadblock. So now the RNA polymerase is going to move to the right across the DNA to the gene 1, gene 2, and gene 3. mRNA is going to be made. RNA polymerase, remember, it copies one side of the DNA and it makes an mRNA strand. The mRNA strand then will leave the nucleus. It'll head out. Actually, there's not a nucleus in a bacteria, but regardless, it'll head out to a ribosome. Although it doesn't say it here, the ribosome then, the tRNA, the trucks, will pull up with their amino acids. The amino acids will join together and you will produce a protein because there are 3 genes, there are 3 mRNA molecules that are created and 3 separate proteins. And those are the proteins that the bacteria needs in order to digest lactose. The RNA polymerase can keep reading that gene over and over and over again and keep producing more and more RNA, mRNA, and more proteins until all of the lactose is gone. At that point, the little lactose molecule, the inducer that's attached to the repressor, it falls off, and that causes lactose to return to its original shape. It snaps back onto the operator and it blocks RNA polymerase from moving back again. So that means that we've produced proteins just for the digestion of lactose, but then once the lactose is all gone, we don't waste energy, or the E. coli doesn't waste energy anymore producing proteins that it doesn't currently need. That's it. Now I've included on the next page a link to a couple of, actually, you know what? I'm going to get rid of those because the videos don't actually work. I'll link it on my Moodle site, so I'm not going to put any other pages on it. That's it for LacOperon. Let me know if you have any questions.